everyone, and I'm Jackie from PolyU. And our project is about developing a bispecific antibody to overcome the osimertinib resistance in NSCLC. And this is my team. I'm Jackie, and I will present first. After that, Amber will talk about our project solution, and following with Sam will talk about our experimental methodology. Finally, Valerie will talk about our IHP activity we carried out. And I would like to take this chance to thank for our supervisor, also the instructor, for guidance and support. Without them, it's difficult to initiate a team and also run this project. So before I start, I would like to talk about what is NSCLC. NSCLC is also called non-small cell lung carcinoma. It's one of the most common and deadly diseases worldwide. 1 in 16 Americans have been diagnosed with this disease, and also it's the leading cause of the cancer in the worldwide. NSCLC is one of the major types of lung cancer. Another type is called the NSCLC, which is small cell lung carcinoma. And NSCLC itself is further subcategorized into 3 subtypes, namely adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. NSCLC, according to statistics, it has a very poor survival rate. Currently, there are several treatments for this disease. For early stage, there is surgery. For advanced stage, there's chemotherapy, target therapy, and also checkpoint inhibitors. And for target therapy, it targets several genetic alterations, including EGFR, ALK, and ROS1. And especially for EGFR, they have the highest possibility among all genetic alterations. So, targeting this gene, there are several commercialized drugs, and this is a 20 years timeline. And you can see the giftinib and the erlotinib are the first generation of drug. However, applying them, there's a problem, which is the drug resistance. And there's a gatekeeper mutation contributed to it, which is called T790M mutation. And after that, scientists, to deal with the question, we invented the third generation of drug, which is called osimertinib. And it's also our project target. However, this drug has a huge hurdle. When we look at the survival rates, we can see that the survival benefits is modest, which means when we compare the placebo group and also the patient receive osimertinib, the survival rate is very similar. And according to statistics, patient can only extend 3 months of life, which is not a very satisfactory number. But the problem is, why is that so? And it's because of the acquired drug resistance. Therefore, there's a great importance to understand the molecular mechanism behind the drug resistance. So what do we do to investigate the drug resistance? We have first trained our osimertinib resistant cell. And this done by, we use a cell line called HCC827, which is a lung cancer cell line. By applying an increasingly dosage of the osimertinib, we have successfully changed the cell from sensitive to osimertinib resistant. And this is verified by the morphology change and also the increased number of IC50. So after that, we have carried out our first dry lab experiment, which is investigating the kinase profile of the resistant cell. And this is achieved by a technique called XO44, and this is kindly provided by our supervisor, Dr. Chow. XO44 is in fact a probe that can bind to 133 kinases to investigate the kinase activity of the cell. And after we apply this probe to the live cell, we carry out the fluorescent gel. And you can see that it visualizes the kinase activity of different proteins. And to quantify this kinase activity, we have carried out another experiment called mass spectrometry. And in this mass spectrometry, you can see 2 clearly results and changes, which is cMET. Kinase activity is highly upregulated, while EGFR activity is downregulated. However, it's logical to know that XO44 is measuring the phosphorylation activity of a protein. However, osimertinib will suppress the activity of the EGFR. So it's a great importance to know that if EGFR is really downregulated or the activity is lower. So we carry out extra experiment called Western blot. And you can see there, the band intensity for osimertinib is much lower, which means much higher, sorry. And then, so we can see that EGFR is actually also upregulated and overexpressed. So this series of experiments draw us a conclusion, which is cMET and EGFR is both upregulated and overexpressed in the osimertinib cell. So we know that cMET and EGFR have been upregulated, so we have a hypothesis, which is by inhibiting both cMET and EGFR at time, we can overcome the osimertinib resistance in NSCLC. So in the following time, I will pass to Amber to talk about our project solution.
Thank you, Jackie, and I'm Amber. Since we assume that if we target to both EGFR and cMET can inhibit the cancer development, so our solution is to develop a novel peptide bispecific antibody, both target to EGFR and cMET simultaneously with a synthetic biology approach. So firstly, I will talk about what is bispecific antibody. As we all know, the nature antibody only targets one antigen, but the artificial bispecific antibody can bind to two different epitopes or antigens. Here are some examples of bispecific antibody. They can be many different forms. And it is already been proved that the bispecific antibody has a potential on cancer immunotherapies, and it has a synergistic effect that is unachievable with a mixture of monospecific antibody. But the bispecific antibody are all artificial products, so the production procedure is complicated and can be very expensive. The financial problem can be a big trouble to cancer patients. So our solution is to make a novel bispecific antibody which can effectively bind two antigens with a lower production cost. Here is our design. There are three main structures. The first one is cMET antibody. We buy it directly from bio companies. And the chemical linker, which is very important to conjugate antibody and peptide. And finally is the cyclized peptide against the EGFR. And how to synthesize this peptide could be the main part of our project. So our solution is to use a bispecific antibody which named PolyU nearby bind on EGFR and cMET simultaneously. Therefore, it can reduce the downstream signals and inhibit cell proliferation. So I will pass to Jem to tell guys how can we synthesize this bispecific antibody.
And thank you, Amber. And this is Sam. I will be in charge to introduce our web lab of how we synthesizing the novel bispecific antibody. More specifically, synthesizing the EGFR binding peptide. So our novel bispecific antibody will target two biomarkers that is upregulated in the lung cancer cell, namely the EGFR and cMET. By targeting the cMET, we'll have the traditional anti-cMET antibody. By targeting the EGFR, we'll synthesizing the EGFR binding peptide through a biosynthetic rate in a PEP24D expression factor. So what this factor is actually contains several parts. First, it will have a original replication that is facilitate the replication of plasmid in the bacteria, and the selectable markers such as the kanamycin resistant gene that will select our recombinant plasmid, and a T7 promoter that will initiate the transcription of our peptide. But most importantly, there's three main parts in our multiple cloning site. First, the gene encoding the EGFR binding peptide. Secondly, the CBD, short for the chitin binding domain, will be used for the purification of our peptide, and the indene flanking at the C and N-terminus of our peptide used for the circularization of our EGFR binding peptide. Initially, we'll have a linear peptide that's shown in this graph, and this is the indene-independent and use his step for purification. However, we find that due to the proteolytic degradation and lower binding affinity, we'll propose a new design with a circularized EGFR binding peptide as our secondary binding, which using the indene-dependent and circularization of our EGFR binding peptide upon the C and N-terminus of our splicing indene. And we'll use the CBD for purification of our circularized peptide. There are total 3 settings for our vector design for synthesizing the cyclic peptide. For the first one, we're using the intin-linked EGFR binding peptide with the CBD as our target setting. Secondly, we'll have the scrambled peptide as our negative control. But we found out that both the first and second setting only accounts for 12 amino acids, which is quite short for peptide. So we'll propose a third setting using the EGFP as our short for the enhanced green fluorescent protein as our positive control to confirm the intin system can actually express our short peptide. Our regular procedure will start with the restriction digestion of our both vector and EGFR binding peptide, and then produce a compatible and ligate into a recombinant plasmid and transform into a top 10 competitor cell for storage. After that, we'll do a sequencing and confirm no mutation. We'll transform into a second host called the BL21 to do the protein expression. After that, we'll do a guiding column to purify our peptide. So after we synthesizing and purify our cyclic EGFR binding peptide, we'll conjugate it into an anti-cMET mono antibody to become our novel bispecific antibody called the PolyU. So the bispecific antibody will targeting the EGFR and cMET like shown in this graph, and further analysis such as the MTT and ELISA will test our novel bispecific antibodies efficacy and binding affinity. And I would like to introduce our next teammate, Valerie, to talk about our IHP of our iGEM team.
Hello, I'm Valerie. Last but not least, I will share on behalf of IHP team what we have done during this period. In this summer, we interviewed 3 clinical oncologists in different hospitals. They are kindly to share with us the recent perspective of the treatment of NSCLC. And their advice inspired us a lot. All interview content are made into broadcast to let more people know about the NSCLC. In terms of public education, we are using the exposure and the sharing potential of social media, like YouTube, Spotify, and Instagram, to increase the public awareness of both our team and NSCLC. And in our podcast and reels, we answer the common questions and concerns about NSCLC. And we're using animated video to raise the awareness of NSCLC among different age groups. For social service, we are honored to cooperate with Cancer Fund to donate money to cancer patients in need for selling the charity T-shirt to the public. And we are conduct an online survey to understand the public awareness and concern and the knowledge of NSCLC. And this data will be used to plan the follow-up activities. In this year, we gained an initial success, and we will try to going on and then keep working on it hard and put our effort into next stage. This is the end of our presentation. Any comments and questions are welcome. And please follow our Instagram and podcast. Thank you.
Because our drug is a pharmacology drug, so before we can commercialize this drug, many steps need to be done. For example, the clinical trial, and before the clinical trial, there is animal model we need to test. But I think it still needs further steps and many money to achieve this.
So we guys can do this project like after the iGEM?
If we get some money from the iGEM or support, probably will do. Yes. And we also join some entrepreneurship competition. There is a competition in our Poly called the Future Health Challenge. We are joining this and going to give a pitch, and if we win this, they will give us $100,000, which can support our future clinical trial study. Yes.
Did I understand wrong that the cMET and EGFR have to be inhibited like simultaneously?
Yes.
So is it possible that you separately express or produce the antibody and the peptide instead of like producing them in one structure?
Yeah. We buy the cMET antibody directly and add EGFR peptide to binding on EGFR receptors. So actually, I show some structures here. There are many different bispecific antibody from other companies. So doesn't matter how the structure is, just we use this structure because it can cause a lower production cost. It's very important.
So can you just apply like the antibody and the peptide simultaneously, but they are not in one structure?
No. Actually, mentioned here, like the bispecific antibody, bispecific antibody has a synergistic effect, which is unachievable with a mixture of monospecific antibody. Like if you only use the EGFR antibody and the cMET antibody, it doesn't work as a bispecific antibody can bind to the same cell.
So someone has tested the...
Yes, it's a conclusion.
Oh, okay, thank you.
Any question? If no, so